invitation. It's really a pleasure to be here, and I want to say that I've never had a seminar publicity piece drafted with so many exclamation points and so much excitement. It actually was really wonderful, and I thought, I can't wait to have the opportunity to talk with the group. And he had told me that it would be in the room a diverse audience with many different interests, and I hope that there'll be something in my talk for everyone in the room today. And I would encourage questions and interaction as we go along. That's what I enjoy most. And more than anything, thank you all for being here today. So I was thinking a little bit about how to frame my talk, and I'm just going to get our slides up here. I can see the mouse. There we go. And, you know, many of us on campus have both teaching and research appointments, right? And so often we find some harmony in our lives when the different pieces come together. So several months ago, I was reviewing the term paper for undergraduates in one of my undergraduate courses. And I had read maybe 50 or 60 papers, right? It was the end of the semester. I was just trying through all of them. I got to one, and the last sentence in the paper, okay, and I made it all the way to the last sentence. I was reading along, and it said, "We are living in the symbiotic renaissance." And I was like, what? What did that person just write in this paper on microbes in the human gut? I was like, what? The symbiotic renaissance, what does that mean? And to the student's credit, he finished it with, dude. And so I think that very much like I had reached a point of exhaustion intellectually in the semester, so too had he. But it made me wonder, what did that student really want to get at with that idea of the symbiotic renaissance, right? And I think that's a really good question. So my background is totally in science. I have an appreciation for history and music and the arts, but I really have a stronger background in biology. So I thought, what do I know about the Renaissance, right? And I thought, well, there was something like this, right? A heavenly presence reached out with his noodly appendage and inspired great change in human society. And I thought, who do I know who are the most important components of the Renaissance? And I thought, well, okay, there are probably 4 artists. But of course, I grew up in the Ninja Turtle generation, so I'm not necessarily in a position to speak to that. So what I thought I would do was go to the Oracle of all knowledge, Wikipedia, and ask, what are traits of the Renaissance? And could be applied to our thinking of symbiotic relationships? So what I realized was that when we speak of the Renaissance, first of all, we're speaking about a period in human history with revelations in philosophy, when classical ideas were cast in a new light. Secondly, the arts, okay, were filled with new perspectives, framing and arising from new techniques. And then finally, even the landscape of human experience changed, and that was in terms of architecture, through the application of newly rediscovered methods that changed what we could see and what we could build. So thinking a little bit about how that translates to symbiosis has actually been a very satisfying process intellectually. When we think about what is referring to a term that was coined by the person pictured here, in the late 1800s, de Berry described symbiosis as the living together of 2 unlike organisms. And from that point, we have moved into a Renaissance period in our thinking, where traditional ideas are cast in the new light of computational biology, where the arts are bringing us a new perspective through tools that we're applying in new ways or that we're developing as we go, and the architecture that is the field of biology is changing as we understand that each one of us harbors every plant we see in the world around us, whether it's in a greenhouse or outside, so too harbors a symbiotic microbiome. And so for me, thinking about how we study symbiosis and what that field is truly a Renaissance phase. And so I wanted to use that to phrase my talk because it's not simply a spectacular time to be studying symbiosis generally. It's also a spectacular time to be studying the symbiotic relationships between perhaps the most beautiful clade of life, fungi, and the organisms with which they interact. And this is just one example of how those interactions manifest into something large and beautiful. These are lichens. Lichens, of course, being the living together in a symbiotic relationship of a photosynthetic partner, an alga, and a fungus. Okay? And so with that background then, I focus on fungal symbioses in my work. And I would like to just give us a brief background about fungi to get us all on the same page, and then we'll move into where I'd like to go with the talk today. So when I say it's a spectacular time to be studying fungal symbioses, this is for a number of reasons. First of all, simply from the perspectives of ecology and evolutionary biology. are getting new insight into the origins and activities of fungi in natural systems. We're also beginning to use new tools to disentangle intricate actions between fungi and other organisms. And one example is between this troglodytid beetle and the fungal symbionts that it harbors in its gut. Those symbionts allow that beetle to eat the wood that it feeds upon to digest it. Without those symbionts, that would not be possible. Okay? Together, these new tools, new initiatives, and new approaches are giving us new insights that go far beyond fungal biology itself. We're thinking about how broadly organisms change, move, and diversify over time, how natural and agricultural systems function and respond to change, how genomes work, both on their own but in symbiotic associations, and then finally we're beginning through the lens of fungi to tap an incredible wealth of biodiversity, much of which has not been examined before. So if we were to draw some boxes here, these would highlight the work that my lab does in general. The sorts of approaches shown in brown and the sorts of exciting directions that we feel are certainly part of a new frontier in blue. So let's talk just a little bit about fungal biodiversity. How many species of fungi are there? So if you were to stop the average fungal biologist on the street, you might get a range of answers. At the moment, there are somewhere between 69,000 and 100,000 species of fungi that have been described. They have a name, they're in a collection, they're formal, everything's there. And the vast majority of these are what we call parasitic macrofungi. They're really big, beautiful ones, the ones that eat mushrooms and rot wood and do stuff like that. So here are a few examples of those macrofungi. But the fungi that get me really excited are microfungi, that is fungi with microscopic fruiting bodies. These are the ones that we affectionately might call the molds. The ones that when I tell my mom I took all that in school to study, she's saying mold, that's so bad. I'm like, it's okay, I understand. And so it turns out that the vast majority of fungi have microscopic fruiting bodies. They're the molds, the small guys. As a whole are thought to be hyperdiverse, and working estimates for the number of fungal species on the planet range from a conservative end at 1.5 million to a probable end at, with most mycologists falling out pretty comfortably at about 5.1 million species. So what's pretty awesome from the side of a mycologist, fungal biologist, is that we have some job security if our job is to go out and discover and describe biodiversity because of that tremendous gap between what we think is out there and what's been described so far. Okay? This also raises a question, though, and that question is, where are the missing fungi, the ones that we think are out there but we haven't described yet? Well, it turns out that most of these fungi are what we call cryptic microfungal symbionts. They're symbionts living in close association with other species, typically plants. They are cryptic in that they're hard to see, and they're microfungi in that they have microscopic fruiting bodies. So when we think a little bit about plant symbiotic microfungi, okay, what we can quickly do is organize all of that biodiversity into a couple of functional groups. So the first are the ones that live on plant surfaces. So these would be parasitic or epiphyllous fungi, them growing on a surface in a tropical forest. They're growing on the surface on the outside. We have pathogens, of course. So fungi disease and major ecological and economic impacts. And then when we go below ground, of course, many of us are familiar with mycorrhizal fungi that are forming associations with roots and aiding plants in nutrient and water uptake. So the group of fungi that I'm going to focus on today is a group that is distinct from these three well-known groups. And for our purposes today, we're going to focus on endophytes. So endophytes are broadly defined as fungi that live within apparently healthy plant tissues without causing overt harm to their host. So the key points are, okay, they live inside, not outside like the epiphyllous and epiphytic fungi. They don't seem to cause harm, so they differ from pathogens. And for the purposes of our focus today, we're going to speak mostly about those that are associated with stems above tissues. So from the mycorrhizae that are in the roots. Okay? So endophytic fungi. It turns out that endophytic fungi have a nice history of study for about the last 110-ish years. And in particular, they've been studied well in cool season grasses. In fact, the poster child of endophyte biology comes from grass systems. So endophytes of cool season grasses are interesting in a bunch of ways. So briefly, they're not very diverse. There are many species, but not a zillion species. They represent only one fungal family. They're vertically transmitted, so they go from maternal grass plant to grass seed to the germinating baby grass plant. They're systemic, so they grow through above ground tissues. They're not occupying only single cells. And they produce a number of important secondary metabolites. So secondary metabolites are compounds made often for defensive purposes, for example. In this case, these compounds tend to be alkaloids, and the alkaloids can have profound impacts on mammals and insects, including humans. In fact, these were first discovered because they could be found in association with seeds, and because the grass plants that grew from those seeds would sicken livestock. And if you go around the world in many different cultures, you can find that are called grass or drunk grass because of their impact on livestock. But that's actually because of the endophyte inside. It's not the grass per se. Okay? This occasionally surfaces in discussion of human history with regard to, for example, the Salem witch trials, whether or not the aberrant behavior by women who were judged to be witches was due to alkaloids coming from the grain that they were grinding to make bread. Okay? So the grass endophyte story is a classic story of endophyte biology. However, it doesn't take us very long to step outside my field site in Hawaii and to recognize that not all plants are grasses. And in fact, it turns out that a lot of the rules, low diversity, systemic growth, vertical transmission, that we know for grass endophytes aren't necessarily true when we look at endophytes of all these other plant lineages. Okay? And so what I'd like to do is just take a moment as an homage to reflect on a figure that I saw when I was a young person and that changed my life. Okay? And it may not change your lives, but hopefully walk away with something interesting too. So what we're looking at here is one of the first papers to categorize endophytes in a non-grass plant. Let me just orient you to what we're seeing here. So each small rectangle, okay, represents a leaf piece 1 millimeter by 2 millimeters in length. There are many small pieces making up each column and multiple columns. Every different pattern is a different fungal species. And what we're looking at here on this millimeters, okay, is the fungal community of endophytes living inside a tropical tree in a rainforest. So from 1 millimeter to the next, in a perfectly healthy leaf, showing no signs of infection, different species, different species, different species. And what's even more striking than that is not only are they different species growing within a millimeter of each other, okay, but often they represent millions of evolution. Yet they have this shared lifestyle of not causing disease and growing inside healthy plant tissue. So when I saw this, I thought, oh God, how can I be thinking about how plants and pathogens interact or plants and insects interact without bearing in mind these cryptic but powerful pieces of biodiversity inside these leaves. So that did change my life, and it's what prompted me to embark on studies that I've pursued since this paper came out. What I've been able to do in the last 20 years or so is to scale up from sort of the individual leaf level and to ask, how are endophytic fungi distributed among plants or at a global scale? What are their evolutionary origins? How did they converge on this lifestyle of not causing disease? How did they get there and why? What do they do in nature if they're associated with all of these plants? And I can tell you that that one tropical leaf depicted in that figure was not alone. Different leaves on the same tree, different trees of distinctive and diverse endophyte communities. So what on earth is going on with that? And then how do they represent or drive the extended phenotype of plants? And what I mean there is that a plant has a phenotype, but given an endophyte and its phenotype can be changed. And so we're really interested in understanding that. So this is probably a bad sign, but I am now to my title slide. And so the title of my talk is Discovering and Applying Microbial Symbionts to Enhance Plant Health and Productivity. And here what we're looking at is a little tiny piece of boreal forest, so forest in the far north. And we're zooming in on this little area where we have a lichen, we have mosses, we have flowering plants occurring in this tiny area. And what I hope will happen by the end of the talk is that you will look at that, strip away all of the distracting green stuff, and imagine the beauty of fungal hyphae, the endophytes that are hidden in there, but also recognize how we can go from that sort of environment through the work that my team and our related teams do to this sort of result. So here what we're looking at are wheat seeds. At the bottom we have wheat seeds in our control. At the top, under the same conditions except treated with an endophyte on the same day, the germination phenotypes that have been enhanced by endophyte treatment. So we can go from communities understanding ecology to applications through the lens of endophyte biology. Okay? So what I'd like to do is just spend a few minutes talking about the work that we've done to look at endophyte distributions around the world. When we started this work, basically, we knew about endophyte distributions in single leaves. And we wanted to know, where are these? How diverse are they? What do they do in different parts of the world? So we're going to start with a little bit of a global perspective, but for the purposes of our talk today, we're going to zoom in mostly in North America with a little bit of a tropical flavor as well. So over the past 20 years, I've been really fortunate to collect endophytic fungi in places marked with stars. And in each of those places, we have done extensive collections, basically going through the process of isolating these fungi and characterizing them. And let's just take a moment to look at sort of the distribution of our sampling from the plant side. So what I'm showing here is just a phylogenetic tree of plant biodiversity. And if you don't know the groups or don't want to read it, that's fine. Knows that we're looking at everything from green algae to flowering plants. And the stars indicate the amount of sampling we've done in each lineage in the places marked on the tree. You can see that we've worked tropical, temperate, and boreal and Arctic environments. And you can see that we've included flowering plants, conifers, ferns and fern-like things, as well as non-vascular plants. Okay? So when we've done that sort of work, we also have made sure, as ecologists, to have a replicated sampling design. So we go into an area, and what we do is we pick microsites, we characterize those environmentally, and then we sample typically 10 plant species in each little microsite. Okay? And just to give you a sense of how this works, basically we take a healthy leaf, we surface sterilize it, we remove fungi or bacteria from the outside, we cut it into little pieces, and we put it on a growth medium. We then wait, and over time, from that little piece, outgrow the fungal hyphae, indication of an endophyte that we then can transfer and maintain as a living collection. We always process our material within about 48 hours of harvesting it. You may be guessing that we move with a mobile sterile lab, and that is correct. So we have mobile laminar flow hoods that are collapsible, and we fly all over the world with them and set up our little lab wherever we go. That MEA is a standard medium. I tell my students it's kind of like non-alcoholic beer jello, and it happens to just support the growth of lots and lots of endophytes. Okay? We always check to see that what we're getting is really from the outside, not the inside, from the inside, not the outside. And last but not least, we keep pictures around for a really long time. Explain why. Here what I'm showing you is how many endophytes, the percent of isolates we get, okay, as a function of how many months little pieces of culture. You can see that in that first month, boom, we get tons of endophytes in culture. But actually what's interesting is that we get a few that show up after 7 or 8 or 9 or 11 months in the lab. Slowly growing, interesting morphology, and often really, really interesting chemistry. These tend to make it worth the process of trying to keep your leaf pieces clean and not drying out and all that stuff in the lab. Okay? So when we go through this process, there's one other thing that I'll draw your attention to on the tree, and that is this little box for green algae at the bottom. So one of the great places where we can find green algae in terrestrial environments is, of course, in the context of lichens. I talked already about how lichens association between a fungus and a photosynthetic partner, typically an alga. It turns out that if you collect a lichen and you surface sterilize it and you cut it up in little pieces, you don't get the fungal partner that makes the lichen growing. You get lots of fungi that look just like endophytes. We call those endolichenic fungi. And they grow in association with a photosynthetic partner in the lichen. Okay? So over the course of our work, we have focused on endophytes as well as endolichenic fungi to discover biodiversity. With our green algal sampling, okay, we've sampled now many, many lichen species in all the sites where we've been able to work. And this is just to convince you that we've sampled lots of images of lichens too. So if you see lots of red on there, then you're seeing exactly what I want you to see. When we look at lichens, we look at the leafy ones, the branchy ones, and the crusty ones. And they all have these great fungi inside. So what has this effort yielded? Beautiful cultures. So each one of these is in pure culture. And to me, it's like looking at a stained glass window in a Renaissance church. I think they're really beautiful. And what's exciting to us is that we estimate that 95% at a minimum are new to science. They're new species that haven't been described before. So that's wonderful. Another nice thing is that we can them as a living collection. This is a voucher collection that lives in my lab in association with the Gilbertson Mycological Herbarium at the University of Arizona, which I curate. And to date, we have more than 60 of these living, the living collection, available for research if anyone would like to come check them out. The problem that we have is that you can't often tell who they are just by looking at their shapes. They don't tend to sporulate very nicely, and there are a bunch of challenges. So we sequence parts of their genomes, usually the telltale barcode parts that are used in lots of fungal taxonomy. Also, we sequence genomes for a number of them. We've sequenced about 250 of them for full genome sequences now. Okay? So what do we do with this collection other than like walk around and admire it? So the first thing is really spectacular, exciting science, which is alpha taxonomy. We're in the slow process of actually describing species. So that hasn't always been in recent years, but it's important for getting these things from a fuzz ball on a plate to being an established part of the literature. So occasionally we get lucky and our fungi fruit in culture. We're able to describe those species. The second is that we ask, what do the fungi do? And here was one of my favorite plants, actually a picture from my dissertation research. This is Theobroma cacao, the chocolate tree, and it's growing in a greenhouse environment in Panama where I've done a lot of my research. What we've been able to do with this tree is to grow it from seed in clean soil in a clean environment so that as it became a little sapling, it had no endophytes in its leaves. We are then able to introduce a pathogen and introduce endophytes and see what happens. So one thing that you're observing is that unlike the endophytes from grasses, these endophytes are horizontally transmitted. You can introduce them and remove them. They're not passed from maternal plant to offspring. So we have an endophyte-free plant. We put in 7 species of endophytes in order to reflect what plants of that age would have in a tropical rainforest. And then we introduced the pathogen. And let me draw your attention to 2 leaves here. Okay. On the left, you have pathogen plus 7 species of endophytes. On the right, pathogen only, in this case a Phytophthora. And what we're showing here is an example of a actually survive when it had only the pathogen. So when we did these saplings or seedlings with the pathogen, typically we lost twice as many leaves as we would if the endophytes were there. Okay, so lots more damage and lots more leaf death. So it's satisfying to do that work in a closed environment, but we're also able to translate that to the field. So we've worked on sustainable chocolate production. There's an interesting historical pattern of chocolate production in the New World tropics where it's typically small landholders who have a little fragment of forest with some chocolate trees separating their little forest fragment from pasture. And so the chocolate trees are a barrier. They're a little bit of a protective edge to the forest, important in conservation. They're also important in chocolate. And so we developed a system where we could grow our endophytes, rice and river water, and then spray them onto developing chocolate fruit and protect those fruits against disease. So it was very low cost. It was done with basically waste material. Spent rice worked just as well as fresh rice with river water. And what was extremely satisfying was increase the yield and increase the probability that that little forest fragment would be conserved. Okay, so another thing that we're really interested in is how do these fungi interact with the leaves? How on earth do they get into them? So a student who just graduated from my group spent countless hours with the scanning electron microscope. And this, too, is a little bit of a vanishing art, but a truly beautiful one. And what she did was that she sprayed endophyte-free tree seedlings with endophyte spores, waited a while, and then imaged the leaves. And that's what we're seeing here. So you can see a fungal spore. We can see fungal hypha, and we can also see the stomatal opening on the leaf. And this is a primary mode for entrance. And just because I think it's so beautiful, indulge me for a moment, we'll just zoom in. It's so awesome. And then we can also zoom back out and see how many of these germinating spores will frequently congregated around stomatal openings, some spreading out, growing on the surface until they find a wound or another stomatal opening and going in. And I should say this work was done in really highly humid conditions, so the little fungi are a little bit confused. They can't find the stomatal openings as effectively as they do when it's dry. Okay. Once they get inside, they grow between cells. They're very different from the pathogen that might penetrate into a cell, and they seem to get along with the ectoplasmic carbon pretty well. So another component of what we do with these fungi is that if I can isolate them, that means anyone can isolate them. It's a really straightforward process. So we love to engage high school students in the lab. And here I'm just showing one of the students. Had I known that Ashley would be here, I'd include a photo of her. And so we partnered with Tucson High Magnet School. I've had a large number of students working in our lab over the past 12 years since I started here at the U of A. And one example of what we do with those students is looking at the ability of these fungi to produce enzymes. So a bunch of these fungi produce cellulases and lignins that are helpful for getting into plant tissue or breaking down plant material. And this is an example. This is a plate with a fungus growing in the middle, and it's basically the fungus growing on cellulose suspended in a medium. And then if we stain that plate, we can see a zone of clearing that's indicative of the fungus breaking down the cellulose. And it's actually turned out to be really interesting because occasionally our high school students yield some amazing results. And this is just one that has stuck with us for a while. So little did you know this would be a high school project talk. I'll just tell you one interesting result. So we looked at endophytes in Cupressus arizonica. This is Arizona cypress. And it turns out that if you take endophytes of the same genotype, but they were isolated from stem tissue, green stem tissue, or more, leafy tissue, those little oppressed leaves, okay, see that they really differ in their ability to break down cellulose. The ones from leaves are really good at it, and the ones from... We don't know why, but we'll come back to this at the end because I think we might know why only recently. So this has led to the development of curriculum for high schools. So we work closely with 2 high schools now and have been able to get large numbers of students with us doing some field ecology, isolating endophytes, and so on. And then finally, we worked extensively with DNA College in this as well. Okay. So with that background, then let's take one more step to think a little bit about the ways that our surveys around the world have started to help us understand a little bit about these fungi. We'll talk about distributions, applications of the endophytes, and then finally some surprises. That's where we'll end. I figure a Friday afternoon surprise is a nice place to end a talk, so we'll go there. Okay, so I'm going to have about 10 slides here with lots of graphs on them. I'm going to walk through them, and hopefully you'll just walk away with a little bit of what we've learned from our work. So I'm going to with the observation of a latitudinal gradient isolation frequency. So what is isolation frequency? That's the percent of those little leaf pieces that gives us an endophyte in culture. And it turns out that as we go across the planet, not all places are created equal. Okay? So the data that I'm going to show you are 66 plant species, site combinations, all major lineages of land plants, 42 plant families, and more than 40,000 little tiny leaf segments in culture, sampling lots of different plant groups. So this is basically what we see. Okay? So when we're in the tropics at low latitude versus when we're in the boreal or Arctic site, high latitude, there's a dramatic difference in the frequency with which we can isolate an endophyte. So high isolation frequency in a tropical rainforest, lower isolation frequency in the desert, low again once we get to the polar deserts or the far, far north. Okay? All right, so there's obviously a lot going on there that isn't just latitude. So we're interested in plant density, that influences how the endophytes are in an environment. Tissue age, so it turns out that older tissues tend to have more of these things. What lineage of plant? Some lineages have more, some have fewer. Okay, and then finally, abiotic factors. So does it need to be more humid? Does it need to have more primary productivity? And so on. Okay, so that's isolation frequency. How many cultures do we get? Now, how about diversity? So diversity, the number of different types we get, right? And so in this situation, I'll show you data for about 8,000 sequence isolates, and this is about 2,000 endophyte species. So let's make the point that we get 8,000 species in culture and have over 2,000 species represented. There's a tremendous amount of biodiversity there. So we might expect diversity to be highest in the tropics, right? Indeed, that's what we see. So once again, we have latitude on the x-axis. We go from high diversity. The measure here is Fisher's alpha. It basically goes from low to high. That's really high. That's much lower, but still not too shabby. So it turns out that even though in that far north environment we don't get much culture, we're still pretty diverse. When we're in the tropics, we get lots of cultures, and they're really diverse. Okay? Once again, there are lots of additional factors that we're interested in. Is the plant native or non-native? How's it been treated? Are insects eating it? Things that we can look at. Okay? All right, so we might ask then, how does this diversity follow the diversity of plants? If you're in a tropical rainforest and you look around, lots of different species all around you. If you're in the far, far north, you might see a willow and a picea tree, and that's going to be about it. Well, it turns out that we can actually ask, does endophyte diversity increase with local plant diversity? And basically, you don't have to look at the details of these graphs to say no. All I want you to walk away with, they're pretty much flat. That is, really diverse doesn't mean anything in terms of diversity of endophytes when we only look at plant diversity as an explanatory factor. And we have plants, and on the left side we have lichens. But what does matter is the good old environment. And so on this next slide, I'm just going to show you another representation of our primary sampling sites in Central America, Northern South America, and North America. And we see mean annual temperature with the color code here. And it turns out that when we're looking at the diversity of endolychenic fungi at the top and endophytes below, there's a strong relationship. In this situation, what we can see is that warmer places tend to have more diverse endophyte communities. Okay? So that actually is pretty interesting for us here in Arizona because we represent a place where naturally many different biological communities come together, and a relatively warm environment, which happens to be typically placing us a little higher than we expect given where we are latitudinally in terms of endophyte diversity. Okay, so we're almost through the graphs. I want to show one more interesting observation, at least something I think is, and that is that we can take our endophyte collection and randomly reach into it and pull out 200 endophytes and ask how many species do we have. Repeat that again. And we plot the results as they're shown here as species accumulation curves. Okay? What that means is that we have 200 isolates in each case across the bottom for tropical forest, temperate forest, and then boreal forest. And what we see is the black line indicating how many species we find. The red lines are confidence interval around that. So what we can see as we go from the tropics to the boreal forest is lots more species in the tropics, right? So that makes sense. Two things that are interesting about that. One, if you were to take the plant diversity in 50 hectares of tropical forest, 50 full hectares, okay, that diversity value, Fisher's alpha of 86, is what you get. If you translate that to 200 cultures that you could grow on a single petri dish, that's the same diversity that we're seeing. So it's petri dish equaling 50 hectares of forest in terms of species diversity. Okay, so the other thing is, I said there were two things. The other thing is that we might have a pattern in phylogenetic diversity. And what do we mean by that? So here we're looking at the diversity of species of endophytes, but those species represent different major evolutionary lineages. So we see lots of evolutionary lineages in the tropics and fewer ones in the far north. Actually, it's worse. So what's amazing is that if we go to the far north, we find a greater phylogenetic diversity, and I'll illustrate that with the next slide. Okay? We can see our pie charts going from the tropics to the boreal forest. Okay? And there are three color codes, gray and white. Black is one fungal class. Gray is another, something called the Dithidiomycetes. The white piece is everything else. And what we can see is that as we get to the farther north, we get lots of evolutionary lineages. So the take-home message is that if we want lots of species, but in a few evolutionary lineages, we go to the tropics. And if we want lots of really different evolutionary lineages and just a few species in each, we go to the north. Okay? And so we've started to understand those distributions. And what that has led us to do is to have just completed sampling around the boreal forest belt. So over the past years, we've been working extensively in boreal forests. These are wonderful for numbers. One is that you get the same groups of plants and lichens in all of the places shown with the pink stars. Second, they're in the far north, which is beautiful. Third, they're full of diverse, phylogenetically diverse endophyte communities. I wanted to tell you that I'm lying just a little bit with the pink star that you can see in Quebec there, because that looks like one sampling event. But let me actually show you what we did. So we traveled by a float plane, and we did a 900-kilometer north-to-south transect, replicated 3 times, and then an east-west 800-kilometer transect, stopping everywhere there's a star. So we'd land on a lake, we'd get out, we'd collect, get back in the plane, fly to the next lake, get out and collect, finally end up at these little bush pilot places, middle of nowhere. The roads end at the 3rd star north, so that's as far north as we went. And we're able to have our little mobile sterile hoods and do all of our isolations there. So just to give you a sense of what we traversed ecologically, we went from the southern extent of black spruce all the way to the on that 900-kilometer transect. And these are our little portals there that you can see that allow us to do this work. And so a lot of this work was led by Jana Uren, who's now a new faculty member in ABE. And I won't be sharing her results today about this amazing work that she has led with Quebec, but I do want to highlight just a couple of the things that we learned. And one is that sometimes we wonder, if we sample and we get some endophytes, how representative is that of the true community? Is that just a snapshot, and if we went back 3 years later, it'd be totally different? So in the little star shown for Alaska there, we actually did exactly that. We sampled in 2008 and 2011. In doing so, we found that the communities were the same over that time period. So it tells us that that snapshot is actually pretty representative, at least over about a 3 to 5 year period. And so let me show you how we depict it. The thing I'm going to do next is something called a non-metric multidimensional scaling graph. That sounds like a lot, but it's not too complicated. Let me show you what it looks like. So we have 2008 shown in red. We have 2011 shown in blue. Each symbol is a community associated with a plant. Lots of endophytes in each symbol. The proximity of the symbols means they're similar to each other, and having blue over means that the communities are not significantly different. So over these two different time periods, we see that the communities were really similar. Okay. So one other thing that we could do, I told you about the boreal forest having all of these plants and lichens that are similar in species composition or genera as we move around. So what we can do is we can sample some beautiful lichens like this little Cladonia, and we could ask the question, do we see differences as we move from one site to another, keeping the host constant? And so I'm going to show you another version of a non-metric multidimensional scaling graph, but here the shapes are colored in to make them easy to see. And so in this case, what we have is that lichen growing in North Carolina, in Florida, in Quebec, and Alaska. And you can see that the communities are completely different. So this is a widespread host that has a distinctive community of endophytes everywhere we go. Okay? So the last piece of this that I want to tell you about is that when we get these communities of endophytes, we can put them into evolutionary frameworks by sequencing parts of their genome. And we can array those as a phylogenetic tree. Now, typically, the piece of the genome we use for fungi isn't very good for big phylogenies because it evolves too fast. It just doesn't work very well. But we've developed a tool that allows us to use that and still get phylogenetic information out. So I'm not going to go into the details of the bioinformatics, but basically we end up with sequences that don't totally align, okay? And we're able to put them into a phylogenetic framework using a tool called a constraint tree. If we do so, we can end up with phylogeny rectangles. They can be shown as triangles, or they can be shown as circles. And we're going to use the circle tree to show you what we see. So let me show you what happens when we take endophytes and we place them into a phylogenetic or evolutionary context. So my students call this the Eye of Sauron because it's so scary. But let me tell you what we're looking at here. This is the species-rich lineage, and you can see that the branches are colored according to where the sample came from. Now what's cool is that this is basically 2,500 endophyte species arrayed as a single figure. And we can see that in some parts there's lots of the same color, in other parts not so much. I just want to show you that with this we can start to test some hypotheses. So you guys will see little blue around the circle. Those are samples that came from the boreal forest. So when we looked, we realized there wasn't a single evolutionary origin of a boreal lineage that then diversified. Instead, we get boreal samples all the way around. And so it suggests that some of the phylogenetic diversity we see in boreal forests is because these lineages colonized moving as sheets have retreated and the plants have moved north as well. Here's the other message though, and this is the other piece of job security that I really appreciate. Basically everywhere we go, we get different endophytes. So here we're just zooming in on two branches of that tree. They're color-coded again. You can see that we see these patterns of local diversification within our study sites. So for example, if we look at the tree on the left, okay, you can see that bright green North Carolina clade, and a Florida clade, and then an Arizona clade, and then an Alaska clade. So this is awesome because basically we know it's going to have distinctive endophytes everywhere we go, lots of diversity, and the potential then for there to be close coevolution with location according to environmental stress between that plant lineage and its fungi. So that has led to some more local interest. And in the past few years, we've embarked on some local sampling that has been really interesting for us. In particular, we've been focusing on endophytes of wild relatives of crop plants. We have, you guys might recognize the mountains there. So this is a little solanum growing as a weed over here, just on the other side, sort of in that degraded field back in the corner of the campus farm. We've been sampling many different families associated, for example, with grass family. We've been working on the cotton family, and so on. And in doing so, we're asking the question, we sample wild relatives of crop plants under different stresses. Do we find distinctive endophyte communities? And can we then use those endophyte communities to help related crop plants grow under similar stress? So we've been looking both at leaves, but also at roots. And let me just show you one of our first observations. These are just a couple of our plants growing under some degree of stress. So this is a little senna growing in heavy clay soil, super cracked, flowering and looking happy as can be. And then we have our little solanum. So this is yet another non-metric multidimensional scaling. And we see that if we look at grass endophytes, okay, like the Poaceae, okay, they don't seem to overlap all that much. But when we add environmental information to this, all of a sudden we can see a pattern. And let me show you what we're seeing here. So as we move from mountains in southern Arizona, central Arizona, and northern Arizona, okay, into transitional grasslands, desert plants, and weed or wild relatives of crops, if we're being a little kinder, weed endophytes. We see this remarkable transition that can be described as a function of and wetter to hotter and drier. Okay? And so we've started to pull the endophytes from the hot, dry environment, and we've started to see, can those help with drought tolerance in focal plants? And in doing so, we've moved now from working only on wild relatives of crop plants to actually working on crop plants. And in doing so, we've also realized perhaps the most important phase in the establishment of crop plants to get those seeds to germinate and have to have them survive in that early phase. So our current method for sampling in this area is a project where basically we're looking for seed-associated fungi. And we have different environments facilitated by the network here at the U of A of agricultural research sites. And so let me just show you the way that this works. On the next slide, you'll be able to zoom in and see what these little tissue things are. They're not actually tissues or Kleenexes. Actually, they're little mesh bags. So these little mesh bags have 50 micro mesh, and then so soil particles and microbes can get in, but arthropods stay out. What we're able to do with that is load them up with seeds, soy, corn, wheat, different varieties, different genotypes, depending on the hypothesis that we're testing, and we deploy them out into the field as bait. We allow them to be colonized by fungi from soil by horizontal transfer. We then bring the bag back in and germinate them. We look for germination phenotypes and microbial communities. And ultimately, our goal is to relate best germination phenotypes to what microbes were there, and then to test that those microbes are actually useful. So I'll just show you a few pictures from our work. This is here at the campus. So we're out putting out our plots. And what's nice is that this is a microbiome project, which means that our plots, the whole area that we need to ask the farm for is usually like 8 square meters, like spread over 4 areas. So it's pretty minorly invasive. Yeah, this is the cotton field right over there. This is in Maricopa, so this is a sorghum field where we put them out. And then we're also working in native and invaded grasslands. So this is down in the Santa Rita experimental range. And then finally, my favorite site, which is the degraded plot right over there. So I'm pretty happy in this photo. And so just to give you a sense, we've worked in all the places shown with the color green or yellow. Green or yellow means so we're doing sampling to see how these communities change over time. And we're continuing to expand this, but one thing that we're excited about is that we can overlay upon this basically soil chemistry or environmental history or cropping history and test a lot of hypotheses about the fungi that we get. So I told you we bring these bags back in, we separate out the different seeds that were co-exposed to all the microbes in the soil. We surface sterilize them and plate them out on plates. We're able to score their germination phenotypes, and then we also isolate microbes. Some of those microbes are associated with seed death, and that's useful to know. others with tiny little seed germination events, and others with spectacular germination. In some cases, we get great germination and no evidence of microbes growing in culture. In that situation, we use a culture-free method, next-generation sequencing, to figure out who's in that beautiful germinating seed. So we've now accumulated almost 4,000 cultures from this work here in Arizona, and a couple of cool observations that I'll just share with you as we close this section. One is that we can put them into those circular phylogenies, right, that circular tree, the Eye of Sauron. And one thing we can see is that different lineages are kind of interesting. So here, the tips of the tree are colored by where the fungus came from. Was it a disturbed site, native grassland, native desert, or an agricultural site? And we can see in the small circles areas where we get lots of diverse fungi. So those attention because they can grow in native environments, they can grow in disturbed sites, they can do really well in seeds, flourishing in agricultural sites. Okay? So we then select fungi that we think are growth enhancers. We do this on the basis of who are they, what phenotype we're associated with, how frequently we find them, and so on. So these are just an example from an undergraduate project in the lab. These were 6 of focal fungi, and this takes us ultimately to the picture that I showed you at the start, which is enhancement of wheat seed germination. And it turns out that with this particular fungus, a fungus that was isolated at the University of Arizona, we're able to not only enhance the rate of germination and the phenotype of those germinants, but also their drought tolerance. So that's been really satisfactory for us. Okay, so just in the last 4 minutes, I want to show you 2 surprises of things that we found. So the first surprise is I told you I was interested in where did these fungi come from? That is, how did they end up not being pathogenic and being helpful to their plants? Well, it turns out that many of them are closely related to pathogenic fungi. Evolutionary history with pathogens, yet to genome sequencing. Okay. The other thing is that it's clear when we look at an evolutionary framework, many of them came from endolycanic fungi that moved into plants. And that's been interesting for us too. So we're trying to understand that process. In the interest of time, I'm not going to make you memorize this tree or test you on it. But what I'll do is just zoom in on the sort of examples of what I just said. So we can integrate not into but trees, and we can recreate or construct the ancestral state of different branches on those trees. And so what we see here is a little lineage that includes lots of pathogens and one endophyte. And when we apply models, then the ancestors are reconstructed as pathogens. And we see the observation that we had a pathogen ancestor yielding it. Okay. We see that a lot on the tree. Okay. Sometimes we have decay fungi yielding an endophyte. And here our endophyte is something called chetomium. Chetomium does everything. It can be pathogen, a saprotroph, or an endophyte. And then finally, we have endolycanic fungi yielding an endophyte. So lots of connection with pathogens and endolycanic fungi, and we're trying to figure out why. So if we map all of those transitions, we have lots of exchange between these 2 states. And obviously, in an applied situation, we don't want to be putting pathogens out there or potential pathogens. So we're really exploring that pretty intensively. And then we also see lots of transitions endolycanic to endophyte. So why these are happening is the subject of work with sequencing full genomes, and then also a lot of really traditional biolog characterization, looking at phenotype. Okay? And the second and closing surprise. So we've talked about how inside lichen plants, right? So it turns out that those fungi themselves have symbionts within them. So that's our world. We're not the first to find them, but we're the first to be looking at them really broadly in endophytes. These additional symbionts are bacteria, and they're bacteria that live inside the fungi themselves. So here, it's a little bit bright in here, but hopefully you can just make out the edges of that fungal. And this is an image that was made by Michelle Hoffman, a former student in the plant pathology group at the University of Arizona. And what she was able to do is differentially stain so that bacteria. So it turns out that lots of fungi have these endohyphal bacteria. And when they live inside the fungi, they do lots of things. So Michelle's work basically was to initiate screens of lots of endophytes. At the time, we had a bunch from Arizona and North Carolina. She found that about 30% of the strains had endohyphal bacteria, It turns out that that's probably more like 80% in the temperate zone once we continue to look with better tools. They're in every major lineage of fungi that we check, and basically everywhere we go. So why is that interesting? Well, let's do this of the bacteria. When we started this work, the only bacteria that were known to grow inside fungi were the blue ones at the top. But every time you see something bold, that's one we found. So we really were able to expand our knowledge of what it takes to grow inside a fungus. We also found that you couldn't predict which bacterium was there by knowing what genus of fungus you had. So there was no clear coevolution between the bacteria and the fungi. And then finally, we looked really carefully at a fungus called 9143 Pestilidiasis. This one is amazing, this yellow, beautiful bacterium called Lidobacter. And when we look at this, what we realize is that we can cure the endophyte of the bacterium. So we can have the fungus plus the bacterium, or the fungus alone, or the bacterium alone, and see what happens. So what surprised us was that apparently my talk is over. What surprised us was that the bacteria really shape the endophyte in culture. So what we see is here a fungus, 9143, that has its bacterium plus or doesn't have the bacterium minus. And we see that when we heat stress that fungus, it grows better when the bacterium is present. The bacterium has thermotolerance on the endophyte. Here we see production of again, in the presence of the bacterium, we get a great production of indole 3-acetic acid, and the fungus is by itself. If we then filtrate from that and apply it to, for example, tomato seedlings, what we see is enhanced growth of tomato seedlings when they're treated with product of fungus plus bacterium relative to fungus alone, relative to just the growth medium of water. So we see a promotion of plant growth with the fungus-bacterium consortium. Okay? And then finally, we can translate this back to endophytes when we're looking in the field. We're able to do small animations. This is, again, showing you my appreciation of and related plants. And what we've done is that we've homogenized endophyte material in water, and we hang that Christmas ornaments on the tips of the branches for 48 hours, allow the endophytes to colonize. And what we see in the graph at the bottom is that typically better colonization relative to when the bacteria is absent. We see that in 3 different plant species, and we can this back out and see that it looks great. What's interesting is that at the top, in C, we have the fungus with the bacterium. In the middle panel, part of C, it's just the fungus by itself. And then R is when we've grown the bacteria, fungus separately, and we've brought them together. So even if they look the same on the plate, they behave differently in nature. So that has been a surprise for us, and we're really exploring how those endohyphal bacteria can influence endophytes and be scaled up to influence plants. So a little bit about bringing the talk together. So what we have done is talked about how basically where and influences how diverse, how abundant, and who endophytes are. Okay? That's influenced by the local environment further. Okay? If we then zoom in on the plant, okay, that of endophytes inside, and on the endophytes, they will have bacteria inside. All of these pieces are connected, and just to get back to the main theme of the symbiotic renaissance, I think that this is part of our changing thinking, that it's not simply a one organism by one organism set of interactions out there in nature, but these complex interactions that are layered in many ways, and that's going to be exciting, I think, for translating fundamental ecological research to applications in controlled environment ag and so on. So with that, I really want to thank you for your attention. You guys stayed with me really well, especially late on a Friday. And I also want to thank my lab. This is my research team from last year. And if you'd like to learn more, you can find arnoldlab.net. Our boreal work is at endobiodiversity.org. And then some of our work that I did about basically the seed experiment is at tropicalseeds.org. And then finally, I mentioned that I'm curator of the mycological herbarium, and that's at the bottom. Okay. That's enough for today. You guys very, very much. Appreciate it.
Wow. This is really not only breathtaking from the point of view of the biology, but the presentation as well. I'm going to open it up to questions, and I am going to ask you to wait for the microphone because people out there in Etherland would like to hear the question as well. Jack, you're first.
Thank you once again. That was a superb presentation.
Thank you.
There are implications here as we look at the climate change and aspects of the heat resistance or the adaptation ability of various grasses. And is it possible that you're looking at one of the most important adaptation mechanisms for survival of the planet as far as plants and those that depend on plants?
What a nice question. If there are any funding agencies listening, absolutely yes. What I would say is, the way we view these fungi and their bacterial associates is very much like what you said, in that we imagine you can instantly manipulate the phenotype of a plant by simply adding something that is not genetic modification and it's not breeding. And so the question that we have to answer before we can make this a broadly applicable tool is how stable is it and can it persist in the environment without promoting undesirable. That's where, at the moment, a lot of energy is being dedicated. But ultimately, we think basically you're giving a plant 1 billion years of R&D in 1 minute when you inoculate it with these endophytes. And so we think that that's an important and probably potentially historically important aspect of plant adaptation to stress. We don't think that these plants would necessarily want these fungi otherwise, because the fungi rely on the plants for carbon. They can, in tropical forests, there's very low light, can be stressful. But if they greatly benefit the plants, then there's a balanced in there. Thank you for that nice question.
Got another one.
Hey, hi, Alon.
So I was wondering if you've seen any of these endohyphobacteria within or in association with monocellular fungi.
With yeast.
Yeah.
Yeah. We have, they have been found in mycorrhizal fungi, so in some of the mushrooms, for example, and they're most famous in our arbuscular mycorrhizal fungi. But as far as having been found in yeasts, not yet. That would be a great study. Yeah.
With your work in Panama, I happen to remember on the human side of things that there was a report 5 or 6 years ago of one part of Panama where the workers had a habit of throwing the leaves into boiling water, and it was sort of like having their iced tea as it was, but there was an unusual human epidemiologic finding that there was a very low incidence of heart disease, hypertension specifically, and then there were other things. And the Mars Candy people got after that, and you may be right on top of all of this information. I think the bottom line question is, were they really looking at the endophytes within the plants being a therapeutic modality?
Great question. So there is growing interest in the power of endophytes as producers of pharmaceutically important secondary metabolites. And our affiliation in parallel here at the U of A is on that question. And so it's been an interesting journey. Okay. Okay. Oh, good. I feel like a rock star. Okay. Yes. Yeah, I can just read the question. Yeah.
In terms of diversity, I believe there was higher diversity of endohyphal fungi in regions with higher mean annual temperature. But have you tested any factors concerning like soil osmotic potential or soil moisture when determining endophyte diversity?
That's a great question. We haven't done that explicitly, and our interest in doing so is motivated by the fact that sometimes we find more diverse endophytes than the local environment would suggest. And so that is relative to the climate of the area, we're getting more interesting endophyte communities than we would predict. And so there may be something interesting about sort of water dynamics in soil and plant water stress. So that is a great area for future research. Absolutely. Thank you. Good idea. One more.
I liked your last representation of the Russian dolls going from plant to endophyte to bacteria, but have you noticed any viral communities or any association unknown that may be attributed to viruses?
Thank you for that question. We finally ran out of hands and resources, so we haven't looked at viruses. But in collaboration with other researchers at the U of A and researchers elsewhere, we are beginning to do some of that work. But what's exciting is that other researchers have found that, for example, thermotolerance can be conferred upon a grass by an endophyte, but only if that endophyte has a virus within it. A particular virus, an interesting virus. And so we think that the bacterial story is only one piece of the complex microbial interactions that are interesting, and that viruses almost certainly are worth looking into. And every time people start looking at these poorly known fungi for viral diversity, there's a tremendous amount of diversity. And we have almost no idea really what the vast majority of them do. That's a great question.
In view of the hour, I'm going to close the formal Q&A session, and I'm sure Betsy will stick around for a few minutes, come up and have a conversation. Thank you very much all for coming, and thank you, Betsy, again.